and was available for people above 18. But by the time we started the trial, there were recommendations to only use it in participants above the age of 13. That's why you see here we had a 2-to-1 randomization then in people above 30 years of age. We did 2 doses, administered 28 days apart, and the primary readout was 2 weeks after the primary series. And specific to this trial, as it was a head-to-head comparison, we did a superiority analysis as the first co-primary endpoint at day 43. And in addition, we were looking at non-inferiority in terms of seroconversion rates, as well as, of course, we were looking at frequency and severity of any adverse events up to that time point. Firstly, coming now to the immunogenicity results. So we could show that it was highly immunogenic across all age groups that were tested. And we met both the co-primary endpoints. So first of all, we could demonstrate superiority in terms of geometric mean antibody titers at day 43, indicated by a GMT ratio of 1.39. And also we were able to demonstrate non-inferiority in terms of seroconversion rates within the well-defined margins that were set for the non-inferiority endpoint. So both vaccine groups were well above 95% seroconversion rates. On the right side, we were also looking at antigen-specific interferon gamma producing T cells, and they were not only induced against the S protein, which of course is predominantly done by the majority of the vaccines, but we were also able to induce interferon gamma producing T cells against the N protein, and to a lesser extent on the right side against the M protein. So that was immunogenicity. Coming to safety. Here we're looking at injection site reactions, so solicited injection site reactions on the left side after 7 days after the first vaccination, on the right side 7 days after the second vaccination. And what you can see here is predominantly pain, tenderness were induced with both vaccines. However, our vaccine induced reduction in solicited injection site reactions after the first vaccination. And when you're looking at the second vaccination, you see a tendency to decrease reaction rates, but there was no difference between both vaccine groups. Looking at the same thing for systemic reactions, so solicited systemic reactions again after the first vaccination and after the second vaccination on the right side, 7 days again. You can see here a similar picture. Predominantly headache, fatigue, and muscle pain were induced with both vaccines. And again, the inactivated vaccine had a more tolerable safety profile compared to the vectored vaccine in that case, so with the reduced reaction rates. And again, what has been demonstrated for the injection site reactions, you can also see after the second vaccination here rates reduced for both treatment groups, and you can see that they are very comparable. What we also did, we were not only looking at humoral immune responses that I indicated previously, but we are also so we were not only looking against homotypic response, but also against heterotypic responses against different variants of concern. And we're doing here pseudovirus neutralization assay in collaboration with our partner at the DPZ in Göttingen in Germany. And what you can see here is that when we're analyzing samples after priming, and in dots you can see it as against the inactivated vaccine, and in squares against the vectored AstraZeneca vaccine, you can see that for the ancestral strain, the B1, you have similar rates elicited. And when you're looking at on the Delta variants or the Omicron, in that case we were looking at BA1 only, you can see that there's a significant better response when looking at the inactivated vaccine. We have done homologous boosters, so in that case the subset of the phase 3 trial that has received the priming with inactivated vaccines, we boosted with the inactivated vaccine again, approximately 8 months after the priming, and we could show that we have a 3.6-fold increase post-immunization levels in antibody titers, as well as approximately 28-fold increase versus the pre-booster titers. And that is indicative of a 90% efficacy against SARS-CoV-2 infection. What we also did, we also then took the samples after booster, and we did the same analysis, and just here keeping in mind, there's no comparison now because of course we were using primary inactivated, and then we were boosting inactivated, so there's no comparator in that case here. But here we are also looking in the pseudovirus neutralization assay, specifically at different Omicron subvariants. And from 18 of the 19 samples had detected neutralizing antibody titers. And we couldn't detect a difference between the ancestral Wuhan strain, which is on the far left, the B1 strain, and the Omicron variants. And when you normalize now, and that's the fold change on the right side, when you normalize now to the Wuhan titers, so the B1, then the titers against the dominating BA5 sublineages, for instance, are approximately threefold lower. I mentioned the homologous boost, but what we also did is we were taking the AstraZeneca primed arms and we're heterologously boosting them with our inactivated vaccine. And data from that, so we could show that we were able to boost AstraZeneca titers and have achieved higher neutralizing antibody titers than pre-boost, again indicated of a 90% vaccine efficacy. And regarding the safety, we could demonstrate that it was similar to that what has been previously observed in the phase 1 study as well as during priming in the phase 3 study. With that data, we received an interim recommendation from the WHO that our booster dose of our inactivated vaccine following a primary vaccination with a vector vaccine, in that case AstraZeneca, can be considered. What we're also doing now at the moment is we're not only looking at AstraZeneca primed and heterologous mixed boost, but we're also looking at a heterologous booster with mRNA vaccines, where we're trying to boost them with our inactivated vaccines, or we're looking at mRNA primed and additional natural infection and boosting with our inactivated vaccines to reflect more what is now occurring in real world outside. With that, I just want to conclude. So in our phase 3 study, as I mentioned, in the head-to-head comparison study, we compared the inactivated vaccine to vector vaccine, and we could show that it was highly immunogenic, well-tolerated, and induced a broad T-cell antibody response. Also homologous as well as heterologous booster doses of the inactivated vaccine induced broadly reactive neutralizing antibodies against the ancestral Wuhan strain in that case, and also variants of concern, including the one that is currently circulating, the BA.4/5 Omicron variant. And we could show that the booster dose, being it homologous or heterologous, had a safety profile that was comparable to the one that we have observed in the phase 1 or in the early phase 3 studies, and it was very well tolerated. With that, I want to end my presentation. I want just to acknowledge the study participants and the clinical trial site and our vendors conducting the studies, our internal Valneva project team and clinical study team members, our DSMB committee, the persons conducting the neutralization assays for the variants of concern at the institute in Germany. Our medical writing assistants was very helpful, and also I just need to acknowledge that the study was funded by Valneva and co-funding from the UK government. And last but not least, I wanted to mention that we have a lunch symposium later on today with 2 excellent speakers, and would be very helpful and thankful if you all could join. Thank you very much.
Thank you. Unfortunately,